It's yours bringing the slides. It just takes a long time. Mine transferred, and then it looks like this. So, did you download it?
Good morning, everybody. Happy Friday.
Happy Friday.
The sun is out, although I kept getting these notices that it was going to rain today, but you know, who knows? Anybody got any fun things planned for the weekend? Like studying?
Yeah.
Yes. Cool. So, do we have any questions before we get started? I had a fun time teaching a little bit about respirometry yesterday. So your group is off the hook because another group decided to use this system that I invested time in. I got to program my new Percival for the first time, which got used all night last night, this morning. So that's pretty cool. This afternoon, we get to have a session on infrared camera use, which is going to be kind of fun. I look forward to those things. It's all things that are not GSCI 170. So there you go. There you go. I get to give them an exam today. So, and then I get to give you guys an exam next week, and I know you're all so looking forward to that. I will start the last topic that we will cover on that exam today and easily get it done. It's only like 40 some slides long, so I'll easily get that done before Friday. So. Anything else that we need to talk about before I get started? One other thing that I do need to bring up is when you are working in either the animal room or if you have to be working in my lab, please don't run off and leave doors propped open when you are not there. This is not something any of you could have done because it requires somebody with a key, which means faculty or staff. But a couple days ago, 2 research labs in the animal room were left unlocked. And that's a little disturbing because my research lab, Haley's research lab, and the animal room were left unlocked. And we have no clue how that happened. It certainly wasn't me. Anyway, just be mindful when you are using those rooms that they stay locked for a purpose, because there's a lot of stuff in there. We only trust, you guys, not anybody beyond you guys, sort of thing. So you kind of get it. So anyway, just be cautious. And if you see anybody who's in there who shouldn't be, you can bring that to my attention as well. So anyway, that was something I was asked to bring to your attention. So there you go. Okay. We will actually take it back. We actually will formally meet for lab next week because I need to go over the doubly labeled water protocol with you guys. And I just won't have another open Thursday before you guys do it, because the following week we do our demo rabbit surgery. This is a meeting that will not go the full time, I promise you that. One thing that you could do for me, which will be really helpful, is to watch the doubly labeled water pre-lab that I have posted. It's a video. Again, it was a video that was made during the pandemic when it was in full force. So there may be some content in there that is specifically pandemic related that you guys can just ignore, but most of the protocol related stuff is still good. It includes a video which I think is going to lack sound, but that's okay. on drawing blood from the tail veins of the rat, which is the best way to do it. The video will actually show them doing venipuncture, but which you guys are not going to do. You're basically going to, use a needle or a lancet and prick a hole in the vein and get it to kind of ooze out, because we don't need to collect that much blood. But you will find it helpful. Okay. And then if you can watch that before next Thursday, and come prepared with any questions that you have, that will speed us up a little bit, and then maybe we won't have to spend quite so much time in lab. One of the key things that does have to happen next Thursday is you guys are going to get a little bit of practice sealing capillary tubes, because when you collect your blood samples from your rats for the labeled water, you'll have to flame seal them to preserve them, and so we'll get a little practice on doing that, just sealing some water in tubes, but we'll talk more about that. And then the actual labeled water experiment will occur in a couple of weeks after that. Okay. All right. So we good? All right. Let's get going. All right, this is where we left off last time. I told you that we wouldn't start this last time, even out of time, because this was going to involve a little bit of a brain teaser. So we're going to start this morning with the brain teaser, and no reason why we shouldn't, because it's Friday and it's sunny outside, and you are all excited to learn this content. So anyway, this here is a study on an interesting organism. It's called a brine fly, and specifically the brine fly larva. The thing that Tim Bradley and his group were doing with these brine fly larvae is, when they were studying some aspects of physiology, they noticed that these larvae, because they live, and actually let me show you, they live in Mono Lake. How many of you are familiar with Mono Lake? Not too many? Well, it's in California, so it's one state to the south, and it's in central-ish California, and it is a highly saline lake, hypersaline lake. And at the time that I made this slide, the salinity of the water was over 8%, which is almost 3 times seawater. And I can almost guarantee you that with the droughts California's been having, that's probably gone up. To put this into perspective, this creates an alkalinity that is very similar to a lot of household cleaners. So you get something like, an Ajax or something like that you use to wipe down your counters, and it's just really toxic stuff. But these flies seem to live in it. At the time I made this slide, the pH was almost 10. Okay, just to kind of tell you just how nasty this environment is just for life. And so they were interested in how these larvae survive these very extreme conditions. And one of the things that they noticed is that the water of Mono Lake has really high concentrations of sulfate ions in it. And what they noticed is that the actual sulfate concentration in most parts of the gut was really, really low, which implies that they are actually absorbing it across the gut. So this sulfate is getting into their tissues. But then the concentration increased dramatically when they got to the hindgut, which suggests what? It's probably in the text. You could probably read it. Actually, it's probably in the next bullet point that I haven't put on yet. So what does this imply? What have we been talking about? Yeah.
Transport.
Transport, yes, yes, nice job.
Thank you.
Yeah, so it implies that this sulfate has been being transported from the tissues into the hindgut for the purpose probably just getting rid of it because it doesn't want it in its system. Okay, so transport is occurring. All right, now because the concentration of sulfate was so high in the hindgut that they hypothesized that it was highly likely that this transport of the sulfate ion was occurring against the concentration gradient, which implies what kind of transport? Active, yes, please say it loud. Be proud that you know the answer to that. Okay, so it was hypothesized that active transport was responsible for the sulfate excretion. Now, you ready for the brain teaser? Don't shake your head, no. Notice how excited I am about all this stuff. Okay. How might we test this hypothesis? You can think back one slide, or actually one slide before I started today, which would have been on Wednesday. Yes.
Is that you find a way to block whatever mechanism is.
And how would you block? Using?
Ice. Oh, I don't know.
What was the last thing we talked about on Wednesday? Yeah, go look at your note. It's okay. Yes. Inhibitors. Yes. The I word. Okay. Remember, one of the points that we made about inhibitors, I remember this. One of the points we made about inhibitors is we can learn a lot about transport by using inhibitors. Okay. So what do we want to inhibit? This is the real brain teaser. So we're going to do an experiment and we're going to use an inhibitor. What is it we want to inhibit? It's too early for a nap. Yes. Okay, so we could get an inhibitor to inhibit the sulfate ion pump. So let's say we do that. And this lowers the amount of sulfate we see accumulating in the colon of the digestive tract. True or false? This clearly demonstrates that active transport has occurred. What do you think, Abby? Does it?
Not necessarily.
Why not?
It doesn't really tell you about the nature of the transport.
So what would be the other option that it could be? Yeah, just inhibiting the transporter does not distinguish between active and passive transport. It just tells you that there's a transporter. Okay. What could we add to this experiment to clearly demonstrate that active transport is occurring? This is the harder one. But I am going to extract it from you because I know you really know. What is required for active transport? Yeah. Okay. You have to, you actually have to be making ATP and using ATP to drive this pump. Right. So knowing that, what would you add to this experiment to clearly demonstrate that active transport is occurring? Yes.
Can you add his membrane pump?
Well, the sulfate, the transport for sulfate ions, if it is if it's an active transporter, would be a membrane pump.
Okay.
So Abby's suggestion, we're already going to inhibit that.
Okay.
We just don't know, just by inhibiting the transporter, whether or not it is actually a membrane pump.
Okay.
Yeah. Okay. Some sort of an ATP inhibitor. Okay. So we would need an inhibitor that would inhibit basically oxidative phosphorylation. Okay. And that's exactly what they did. 2 inhibitors. So here's the control. So we're plotting sulfate concentration on this axis here, our various experiments down here. Here's the control. We get the high concentration of sulfate in the hindgut right here. Here are the results with transport inhibitors. It reduced it to almost zero, which means that they were no longer pumping this into the hindgut. But then here are some oxidative metabolism inhibitors as well, which gives you the same result. So the combination of these 2 things, they were able to use these data to demonstrate that transport of sulfate into the hindgut was actually active transport. Does that make sense? Something like this makes a wonderful essay question, don't you think? Questions? Yeah.
So they had to inhibit both the pump and.
No, they actually did them in separate experiments. So these 2 different inhibitors were done independently. So they got this result just by inhibiting the pump. Then they got a similar result not by inhibiting the pump, but just by locally inhibiting ATP production. Yeah, because if they did it together, you really wouldn't be able to separate which one was causing the effect. So yeah.
Yeah, so you had said, I just want to make sure, so you said earlier that just inhibiting the transport makes it a passive.
No, it means you can't distinguish between passive and active. You can only determine that a transporter is involved in moving.
So you, I don't know what I was talking about, so you have to inhibit pump or the ATP production?
Well, what they did 2 separate experiments. They inhibited the pump and showed that once they did that, there was no transport of sulfate into the hindgut. Then they did a second experiment where they inhibited oxidative metabolism, ATP production, and then also demonstrated that when you did that, there was also no transport of sulfate into the hindgut. So by getting the same result with both those experiments, they were able to determine that the transport of sulfate into the hindgut was active transport. Does that make sense? Yeah.
With this, did they use like a short amount of time that this like cyanide isn't just going to kill the cells?
I am absolutely positive it was a very localized experiment.
Okay.
Yeah, because it's been a while since I read the paper, but yeah, they probably were looking at transport, and they were probably just infusing this and just watching the immediate effect before it could get out and spread to a bunch of other things. I guarantee you that an editor or that a reviewer of a paper would have asked that very same question. That's what I thought about when I first saw this. So yeah.
No question. So if the transporter inhibitor is like decreased sulfate concentration, but what about if oxidative metabolism inhibitors had a high concentration? What would that mean? So
I'm not understanding.
there are 2 parts of the experiment.
Mhm.
They did 2 inhibitors. If one of the inhibitors did not show a decreased solute concentration, or sulfate concentration,
No, they both did.
but what if they didn't?
What if they didn't? So let's say that they did the oxidative metabolism inhibitor and you still got a high concentration of sulfate. What would that mean? Huh? It would mean what?
Passive.
It's passive. It's a passive transport. Guys are asking good questions. Anything else? All right. Okay, now let's get ourselves set up to move on to starting to have several topics that are going to deal with the nervous system. And the last issue with regard to membrane transport that we want to deal with is the whole concept of ion gradients, or what we refer to as electrochemical gradients. And the dynamics by which ions move across membranes is really fascinating and really important to several aspects of physiology. We've actually already seen or already talked a little bit about how the energy that's stored in these gradients can be used to drive other processes. You remember secondary active transport. And so ion gradients do a lot of very, very important things. And as we mentioned previously, they're critical for the functionality of the nervous system. The amount of energy that's stored in an ion gradient is dependent on how big the gradient is. So yeah, if you have a huge gradient, then you have a lot of stored energy. If you have a small gradient, you'll have a small amount of stored energy. And then this energy is released, and you can think of this sort of like the conversion of potential to kinetic energy. Energy is released when the ions are allowed to flow passively down their concentration gradient. I think we mentioned this last time. This is exactly how we get ATP production, because the whole purpose of the electron transport system is to establish a proton gradient that's used, and the energy that's stored in that is used to kick out ATP. All right, so we've talked extensively about concentration gradients, i.e. the chemical gradients that drive transport. But when we're talking about ions, there is another driving force that can result in the movement of ions, and these are electrical gradients. So if you have a charge difference across the membrane, that difference in charge will also drive the movement of charged particles. Because remember, opposite charges attract. So an electrical gradient will occur whenever you have a charge difference across a membrane. And remember, last time we talked about the sodium potassium ATPase, most every cell in your body is going to have a potential difference across the membrane. It's going to have a difference in charge across the membrane. There are like no exceptions to that I am aware of. So all cells normally in their resting state will have a potential difference. Okay, I just said that. Ions will always move towards regions of the opposite charge. Okay, so, and this is an important statement right here. When we are trying to understand how ions are distributed on one side of the membrane versus the other, it's always going to be a combination of the effects of a chemical gradient and an electrical gradient. It's going to be an integrated effect. And what that means is the number of ion particles on two sides of the membrane are rarely equal because of this joint effect in setting their distributions. And I'll give you an example of this. So let's say we have a chamber here, and we divide the chamber by a semi-permeable membrane. This membrane is, or on one side of the membrane, you're going to stick a potassium chloride solution. The other side has pure water. And let's say this membrane is permeable to potassium chloride. So as we learned in talking about simple diffusion, if the membrane is permeable to potassium chloride, then over time, the concentrations of potassium chloride on either side of the membrane is going to become equal, right? All right, so let's change this up a little bit. Now, let's add a new ion over here. Okay. So we're going to add a new negative ion to the left side over here. And everything that we said about in the previous slide is the same, except this membrane is not permeable to this new negative ion right here. So what's going to happen is this is going to create unequal charges on the left and right side of the membrane. The left, I'll talk to me later. The left side of this membrane is going to become negative with respect to the right side of the membrane. And so what's going to happen is that will start to pull the potassium back to the left side and repel chloride towards the right side. Does that make sense? And so when this system reaches equilibrium, the equilibrium will result in having a higher concentration of potassium on the left than on the right, and a lower concentration of chloride on the left than on the right, in order to establish what we call an electrochemical equilibrium. So again, the integrated effects of both the chemical and the electrical gradient. Does that make sense? Do you have questions? By the way, we refer to this type of an equilibrium as a Donan equilibrium. Did they ever teach you that in chemistry? Maybe they don't even use that term anymore, but in my era they did. Yeah.
So I have a question. So the potassium, like the KCl, that's the, hold on. Oh no. The electrochemical, oh no. So the chemical gradient, that's what I'm going. The chemical gradient is the potassium chloride, and the electrical gradient is just the charge difference between the two.
Correct.
Okay, that's what I'm saying.
Correct.
Thank you.
So you have two things that are working to set the distribution of solute particles on either side of the membrane. The chemical gradient, okay, which if that's the only thing that was playing a role, and frankly, if this membrane were permeable to this new ion, you would get equal distributions of everything on either side. Okay, but because the membrane's not permeable to this, you have an excess of negative charge over here, so that electrical gradient starts to play a role in the distribution of the solute particles. Does that make sense?
Yes.
Okay. And to be honest, this is what most cells look like. They do not have equal amounts of ions on either side in their resting equilibrium state. And that's because the membrane is going to be permeable to some ions, but not to others. But all ions are going to contribute to the charge difference across a membrane. Any other questions? Yeah.
So, the movement. If you have a semi-permeable membrane, the movement of the ion is dependent on the ion identity, but not necessarily the charge.
Well, I mean, it's going to be dependent on the permeability of the membrane, what it's permeable to. In this case, it's permeable to potassium and chloride, but not to whatever negative ion A is. Okay, this is stuck on this side of the membrane. So as ions move across the membrane due to chemical gradients, you start to enhance a charge difference between these two, and the charge difference becomes significant enough where it starts pulling positive ions back. Okay. And so you end up with that unequal distribution because you have both of those factors. It's kind of a tug of war. In many ways, it's a lot like the two functions or the two factors that drive osmosis. So you have the osmotic gradient and you have the hydrostatic pressure gradient, same sort of thing. Good to move on? All right. So I'm probably beating a dead horse about this, but electrical gradients are huge in physiology. And, you know, we'll spend multiple topics on different aspects of the nervous system, and you'll see that pretty clearly. So as I mentioned, pretty much every cell has a potential difference across their membrane. And in the resting state, that potential difference is what we refer to as a resting potential. So these potential difference, and another name that you'll see for the potential difference across the membrane is just the term membrane potential. It's going to involve passive processes, which are processes associated with what I referred to as a Donan equilibrium in the previous slide, as well as active processes, membrane pumps, so the sodium potassium pump. That's a big player in creating membrane potentials. Okay. The basis of nerve function is all about rapid changes in membrane potential, moving cells off of their resting potentials. Okay, you get these gated ion channels, and these are electrically gated ion channels, and we'll talk more about those later, that allow this rapid flow of ions across a membrane. That's the whole basis of how the nervous system works. We've already mentioned the establishment of proton gradients in cellular respiration. It's another example of a pretty huge difference in membrane potential. And this here is just a diagram which illustrates the variety of transporters that we've talked about in this topic. This is my last slide of this topic. Okay, so we have primary active transport, we have secondary active transport, we have facilitated diffusion, which involves passive transport using carrier molecules, all these ion channels right here. It doesn't list simple diffusion, but that does happen in some things as well. I found this on the web. Boy, you know, the Apple lady is just really active today, isn't she? Get lost. All right, and that does it for this topic. Any questions before we move on and start talking about excitable cells? All right, topic 4, tissue excitation. We are going to start a journey that's going to take us a few weeks to talk about things involved with the nervous system. The nervous system is, you know, it's a big driver of much of physiology, so it's appropriate to spend some time on it. So yes.
Sorry, is this like topic, is it going to be on the exam, or is it going to be on
This one will, yes, I will absolutely get done with this. This is a pretty short topic, so I'll get done with this easily by Wednesday.
Okay, sounds good, thanks.
All right. So cells associated with both the nervous and muscular system and a few other odd systems or odd types of cells out there fall into a category of what we call excitable cells. In other words, they can transmit or respond to electrochemical stimulation. Okay. So we're going to primarily focus on the nervous system with this regard, but there are several of the principles that we are going to talk about with regard to the nervous system, and specifically action potentials, which we will talk about at the end of this topic, are characteristic of a lot of different cells that are outside of the nervous system. The two big ones, of course, are the muscular system and the sensory system. Excitability is based on the movement of charged particles, i.e. ions, across the membrane. And the whole dynamics that are associated with these ion currents are really the basis of how cells communicate electrically to one another. How information is processed in most living things. And as you shall see, the amount that we really understand about this process, although when you go and read the literature, it can be kind of overwhelming. The amount that we've really learned about it is it's a relatively small amount. What you'll see is what we've learned is how individual cells work. We understand that really well. How things like nerves work collectively, we understand that very poorly. So the nervous system, in addition to neurons, nerve cells, consists of the nerve cells plus a lot of supportive cells that we refer to as glial cells or neuroglia. There are actually a lot of these cells. The purpose of these cells is to collect, process, and analyze information. And they coordinate a whole heck of a lot of physiology. And this is what I just said a moment ago. We don't really know a ton about how the nervous system works as a collective unit. And our best understanding is literally of simple circuits. You know, so like if you want to know how, say, like a reflex arc works, you know, we have a pretty decent understanding of that. But, you know, if you want to know some of the details associated with things like memory, we really have a very poor understanding of that. We've done a lot of work on individual nerve cells. In fact, you know, something that will come up in multiple topics from time to time is work that's been done on these giant nerves that we find in squid. They have these huge giant axons that actually are easy to see and easy to work with. And then one of the points that I'll make a little bit later in this topic is that regardless of what organism you look at, nerves function the same way. There is like very little variation in the functionality of a neuron between a squid and a human being. So they're very useful in trying to understand just how they work. So neuron function is essentially the same in all animals. Although neurons really process a lot of complex information, really the basis of how they work is pretty simple. There really are just a few things that are going on. The complexity of the function of the nervous system is in the collective functioning of all these neurons. And how signals from individual neurons can be integrated in order to make a decision. That's where the complexity comes in. So the complexity is in the design. So when you look at things like measuring intelligence by brain size, well, you know, and to some degree that does work. You consider a large brain has a whole bunch more neurons that make a whole lot more interconnections than, you know, something like, you know, an insect brain does. But even an insect brain does a lot of pretty complex things. All right, a little neural anatomy here. The cell body of a nerve we call the soma. And it's actually responsible not only for all the basic cellular things that cells do, but it's also responsible for the integration of incoming signals to the nerve. Signals will be transmitted to the nerve cell through these dendrites. The dendrites extend from the cell. They're generally very highly branched, which means they make a lot of connections to a lot of other cells. And they are going to bring inputs. They are going to input information into the nerve. And this input is going to come from multiple sources. And later on, we'll see at least at a low level how this integration of information occurs. The axon is an extension that comes away from the cell that is going to carry information away from the soma. Axons can be very short. And when we look at nerve cells that are associated with the brain, they often have very short axons. Or they can be very, very long. So if you look at some of the motor neurons that will service like the muscles down our legs, you can actually have axons that are meters in length. When we use the term nerve, we have to have a clear understanding what we're referring to. Often when we call something a nerve, it is in reality more like a cable of nerves. So this here is a cross section of a nerve bundle that when you look in an organism might appear as a single long nerve. But in reality, if it's some sort of like a telephone cable, it's got a whole bunch of different wires in it. And so in cross section, you can see that each one of these circles in here is basically the axon of a single nerve. And the white area there, as you're seeing, is the myelin sheath that's around the axon. A nerve that you all are going to become intimately familiar with is the vagus nerve. The vagus nerve is just like this. It's a major autonomic nerve of the parasympathetic nervous system, and it carries a lot of individual axons in it. Okay. So a lot of times when we refer to nerves, we're actually referring to these nerve bundles. So you need to be clear about what you're talking about. And myelin sheaths, as I think we mentioned before, are structures that that a glial cell will put around an axon that help to speed up the transmission of nerve signals, and we will learn how that happens. There is no tissue in the mammalian body that is more sensitive to damage than nerve cells. And when a nerve cell gets damaged, it can degenerate really quickly. And regeneration is hard. The place in mammals, and that includes human, where you're most likely to get some regeneration is out in the peripheral tissues, so fingers and toes mostly. What's interesting is when you look at cold-blooded vertebrates, amphibians, they tend to exhibit much better regenerative powers when it comes to the nervous system than mammals and birds do. And so amphibians and some reptiles have been involved in huge studies of regeneration, trying to understand how that works in an effort to figure out whether or not you can drive these sorts of mechanisms in humans mostly to regenerate nerve tissue, which would be huge for people who have like spinal cord injuries. Regeneration in peripheral tissues is typically going to occur if you get a severed nerve and the endings are fairly close to each other. They will generally have a predefined path that they can grow and reconnect, and that can happen. But if the separation is too far, then what you typically will have happen is the nerve will start to grow in the wrong direction. And some people even end up getting sensation in areas that a nerve was not intended to provide sensation to. So the nervous system is just kind of an odd thing. So, for years, we thought that nerve tissue, we just didn't grow a lot of nerve tissue, basically as adults. But in reality, there are a few places where we get rather consistent regeneration of nerve tissues. Some studies have been done on this. Stem cells are what you would regenerate brain cells from. And anybody know what a stem cell is? Go ahead.
Undifferentiated cell.
Yeah, it's a totally undifferentiated cell. This is why they're such a hot topic for research, because they literally have not received their genetic programming yet. And presumably you could turn them into anything you want, you know? And that's why there's such high interest in them. In cockroaches, okay, they have a brain region that's called this mushroom body right here. And this mushroom body will contain these, the type of stem cell they refer to as neuroblasts that will produce new, what we call interneurons. Interneurons is the type of a nerve cell that we find extensively in brains. They transmit, relay information, they do a lot of processing. This occurs in humans as well. There are two places, a region that's called the subventricular zone, which is below the ventricles of the brain. So you can see right here in this view, right here in this view, and the hippocampus, which is located right here. Again, stem cells that consistently undergo neurogenesis to create and replace interneurons in the brain. So there is some regeneration of nerve cells in some places, particularly in the central nervous system. Okay, so dendrites are going to be connected in some manner to axons from other nerve cells. So these axons here will connect, if they're going to connect to another nerve cell, connect to the dendrites. All right, and this would be involved in the relay of information from one nerve cell to the next. And then the nerve cell that receives that information, things will go on inside that nerve cell that determine whether or not that information is going to continue to be transmitted down the road. And again, we'll talk about how that works in a later topic. So the signal comes in to the nerve cell through the dendrites, enters the soma. Now, we've already mentioned that the key thing that has to occur in order for information to be transmitted from one nerve cell to the next, or there's this thing we call an action potential. Action potentials get transmitted down, excuse me, the axon here. But what's important to understand is that action potentials do not occur in all the membranes of a nerve cell. You do not see them occurring in the soma. You do not see them occurring in the dendrites. An action potential cannot occur until you reach this region of interface between the soma and the axon that we refer to as the spike initiating zone. The region itself is referred to as the axon hillock, or some texts will call it the initial segment. And so you need to have a signal that's strong enough to initiate an action potential here in order to transmit it down to the axon terminals, the end of the axon. And again, many axons that must transmit information quickly are covered by myelin sheaths because that increases the speed of signal transmission. You will see myelinated nerves in fibers that are going out to target tissues a lot. And we'll talk about how that works later. All right. So nerve function is going to be dependent on the properties of the cell membrane. So we talked a lot about passive electrical processes. So all of these passive transport mechanisms, ion channels, those sorts of things. Okay. Even electrical gradients themselves are passive, are passive properties. All cells have. All cells are going to have passive electrical properties. The other thing that is going to be important for excitable cells are active electrical properties. Okay, and I'll finish up with this today. So active electrical properties are going to involve properties that allow the membrane to respond to a stimulation. These are going to involve these special integral proteins that are called voltage gated ion channels. So we talked about gated channels in the last topic. This is a type of an ion channel that's gated and will be ungated when the membrane potential reaches a certain level. So it responds to an electrical change, a stimulation. All right. Really important for action potentials. Key thing is, and burn this into your brains, a cell cannot be excitable. In other words, it cannot undergo an action potential unless the membrane has active electrical properties. If the membrane has no voltage gated ion channels, then an action potential cannot occur. Remember, all cells will have passive electrical properties. They will all have a potential difference across their membrane. They will all exhibit a resting potential. But only cells whose membranes exhibit these active electrical properties, i.e. the presence of voltage gated ion channels, have the capacity to respond to stimulation. Really important for you to remember that. Okay. All right, I will stop there. Questions? Was that my alarm? To tell me I'm done?
It's an alarm for Donut Friday.
Okay. All right. Everybody have a good weekend.